Hi, Micro Geniuses. This is Brian. I am sharing a screen with you. This is just a PowerPoint, a RESTful screen that you can perhaps look at. Most of this is going to be audio. I might refer to PowerPoint, but this is our video review for the final exam. So I just wanted to give you a review of the different questions on the final. It will be helpful now for you to stop this video and get your old exams or get the key to exam 3 that I put on Moodle. So the key is out there and you can refer to that. The questions on the final exam primarily come from the old exams, exam 1, 2, and 3, and there will be a few new questions based on the lecture materials that I covered last week. So anyhow, make sure that you've got those in front of you, and then we'll get started. Okay, in terms of questions that are going to be on the new material, I'll do those first, and then I'll do the review questions last. The final exam, you'll have 2 hours to do. It will be posted on Tuesday at 8 a.m. and you'll have until Tuesday at 5 p.m. to complete it. So make sure you start it by 3 p.m. so it doesn't time out on you. The final exam has 75 multiple choice true false questions, and it's worth a total of 150 points. And it will be tabulated quickly. I've finally caught up on grading. So this will tabulate very quickly, and you'll be able to get your final grade relatively quickly. The only things that I need to grade on top of that are lab 25, which is due on Friday, and lab 45, which is due on Monday. Okay, now actually, no, lab 45 is due at the final exam. So anyhow, if you get that in on Tuesday, that's fine. Okay, so lesions that you should know what kind of lesions come from meningococcal meningitis, and these are red and purple blotches on the skin. And this goes back to when we were studying the skin and the eyes, although this is an infectious disease of the nervous system. Okay, so these red and purple blotches are called petechiae, P-E-T-E-C-H-I-A-E. Okay, so petechiae are characteristic to meningococcal meningitis, not to be confused with purpura, papule, macule, or you can also get ecchymosis, but ecchymosis is more of a necrosis of the skin where the skin actually blackens. Okay, so next we wanted to talk about coronavirus. So we got a few questions on the final on COVID-19. Okay, you should know that the transmission frequency of COVID-19 has been determined to be 2.0. That basically means that for every individual that gets it, they're transmitting it to 2 more people. Okay, so we're trying to drive those numbers down. And when you hear people saying we need to flatten the curve, that is due to that high transmissibility. Okay. You also should know that the theory that the COVID-19 virus co-migrated into a bat, and that you had 2 different types of viruses that co-migrated into a bat, and that resulted into COVID-19. That which is infective in humans, that's an example of antigenic shift. Okay, it's an RNA virus like the influenza virus, and it can actually get the RNA donated from similar types of viruses. And when that happens, then you get a unique virus that may have not been infective to humans before, but then is infective afterwards. Okay, You should also know that COVID-19, another name for COVID-19 is SARS-CoV-2, because it's related to the serious acquired respiratory syndrome virus that was isolated back in 2002. Okay, so COVID-19 is an example of a single-stranded RNA virus. It would actually say positive sense RNA virus, which means that it can be directly translated, but all you need to know for the test is it's a single-stranded RNA virus. Okay, and those are the new questions that are not review. Okay, and so now I'll go through the questions that are review. Okay, so the first question is, you should know what the valid modes of action are for an antibiotic. So for an antibiotic, you can inhibit protein synthesis, you can inhibit cell wall synthesis, you can inhibit cell membrane synthesis, you can inhibit DNA or RNA synthesis, or you can inhibit folic acid synthesis. Okay, also you should know the different types of media. So the different types of media, you have all-purpose growth media, then you have fastidious media that contains special requirements that are needed to grow bacteria that need certain additives like amino acids. You have enumeration media that is used to count bacteria. You have anaerobic media that actually has a chemical that scavenges oxygen from the medium. And then you have transport medium that is used when you need to store bacteria for a long period of time. Okay, you should know the modes of action of the different antibiotics. You should know that penicillin is a cell wall disruptor. Cephalosporin is also a cell wall disruptor. Sulfonamides are also called sulfadrugs. They are folic acid synthesis inhibitors. Rifampicin inhibits nucleic acid synthesis, and then polymyxin inhibits cell membrane synthesis. Whoops, didn't mean to do that. Okay. Okay, you should know that bacteria have plasmids as well as chromosomes, and you should know that a plasmid is a small circle of DNA that is transferred during conjugation. You should know the individual who is responsible for the use of the microscope for microbiology. That is Leeuwenhoek. You should know that AZT is an antiviral drug that blocks nucleic acid synthesis. You should know what a parasite is. A parasite actually feeds on a living host. You should know what a saprobe is. A saprobe feeds on a dead host. And then you should know the difference between photo and chemotrophs and auto and heterotrophs. Photo and chemo deals with where they get their energy. Auto and hetero deals with where they get their carbon. You should know the factors in the germicidal activity of chemicals. You should know that that includes the concentration of the germicide, how long the germicide is exposed to the contaminant, the degree of contamination, and the nature of the material being treated. Okay, you should know the following structures on microorganisms. Fimbriae are spikes that you find outside of the bacterial cell that are basically cell Velcro. They allow the cell to attach to surfaces. Pili are used for conjugation in gram-negative bacteria, and pili are where the plasmids are exchanged. Glycocalyx is a layer of polysaccharide outside of the cell wall, and that could be a slime layer or a capsule. Cilia are like flagella, but they're more numerous. okay? And they beat in an undulating fashion to give the cell motility. And then flagella are single or multiple, and they are long appendages that in bacteria move in a propeller fashion, and in eukaryotes, they move in a whip fashion. You should know that some bacteria have no cell walls. That would be like Mycobacterium or Mycoplasma. They have no cell walls. So some prokaryotic cells do not have cell walls. Okay, you should know that true pathogens will create disease in healthy individuals, and opportunistic pathogens create disease in sick individuals. You should know that Pasteur was the person who introduced the germ theory of disease. Should also know the different types of extremophiles that live in extreme environments. Barophiles live in high pressure. Halophiles live in high salt concentration. Mesophiles in medium temperature. Psychrophiles in cold temperature. And then thermophiles in very hot temperature. Should know that also the difference between selective and differential media. Selective media actually inhibits the growth of certain organisms over and against the growth of others. And differential media allows everything to grow up, but it gives the colony specific features so you can differentiate between different species of bacteria. You should know how selection pressure works in natural selection, that selection pressure will favor organisms that are more favorable under a given environmental condition or environmental change. Has nothing to do with the complexity of the organism. Should know the different alternatives for sterilization. Desiccation would just be drying. Filtration you use to filter out microorganisms, and that's for liquids where you don't want the liquid to lose flavor. You can use radiation, gamma radiation or X radiation. You can use wet heat, would be autoclaving. You can use dry heat, which would be putting in an oven, which is less efficient. And then you could use incineration. You can just burn something. If it has the ability to be scorched by a flame, then incineration is the most effective method for sterilization. Okay, you should know what commensalism is, and symbionism is synergism. Commensalism is when one organism is favored, but the other organism doesn't receive any benefit or harm. Synergism is when they both receive benefit. Okay. Antagonism is when they are competing for the same source of nutrition. And parasitism is when one is benefited and then the host is harmed. You should know that a simple stain uses one dye and a differential stain uses two dyes. You should also know the difference between an exotoxin and an endotoxin. An endotoxin is a toxin that is shed from a bacterial cell wall, specifically gram-negative bacteria. And an exotoxin is actively secreted from inside the cell. You should know which types of organisms and which types of infective agents are most resistant to sterilization. That would be prions. You should know what prions are. They are just proteins that are misfolded, and they propagate by transmitting a misfolded state. You should know what phylogeny is and what the phylogenetic tree is. And when you have two organisms that are close together on the phylogenetic tree, that just means they're genetically similar. It doesn't necessarily mean that they're morphologically similar. You should know the different parts of the microscope. You should know that the iris diaphragm will narrow the field of view. You should know the ocular is the eyepiece, and the objective is the lens that directly interacts with the sample. And you should know that the condenser actually focuses light directly onto the sample. Should know that the treatment for malaria is quinine. And you should know that gamma radiation, X radiation, ultraviolet radiation, and fluoroquinolones all adversely affect DNA. You should know what lipopolysaccharide is. Lipopolysaccharide is just a gram-negative bacteria's outer layer, which is pathogenic. Okay, lipopolysaccharide that causes fever and shock in humans. You should know that dermatophytes are fungi that infect the skin. Should know the difference between a host and a pathogen. You should know the different terminal electron acceptors for different types of bacteria. For aerobic bacteria, it's oxygen. For denitrifying bacteria, it's nitrate. For sulfate-reducing bacteria, it's sulfate. For iron-reducing bacteria, it's iron 3+. And for methanogenic bacteria, it's carbon dioxide. You should know that a gram-positive cell wall is thicker than a gram-negative cell wall. That's the basis for gram staining. You should know what happens when a flagellum rotates counterclockwise. That results in a run. So the bacteria goes straight forward. If a flagellum rotates clockwise, then that results in a tumble. So the bacteria tumbles end over end. Should know what a carrier is. Carrier is somebody that inconspicuously shelters a pathogen. Okay. Should know what an epidemic is. That's when the frequency of a disease exceeds its normal amount. A pandemic is just an epidemic on multiple continents. Endemic means that a disease stays at a certain frequency and is associated with a certain area. Sporadic means that a disease just pops up occasionally. You should know that in DNA structure, bacteria genes do not have introns. However, archaeal genes and eukaryotic genes also have introns. So the only type of cell that doesn't have an intron is a bacterial cell. Should know what a niche is. The niche is just a role that a certain organism plays in the community. Should know what habitat is. That's where they live. You should know what biomes are. Biomes are just areas that are associated with a certain altitude and latitude. You should know that acetogenic bacteria provide sulfate. Okay, they actually take sulfide from the soil and they convert it into sulfate, and that provides nutrition for plants. Should know the different zones of soil. The rhizosphere is the plant root zone. The topsoil is generally aerobic. The vadose is the zone of soil that starts from the soil surface down to where groundwater starts, and it's unsaturated with water. Mycorrhiza are just part of the rhizosphere, and they're just mold and fungus that allow the plants to acquire water and nutrition. And then the saturated zone is actually groundwater, where all the pore spaces in the soil are saturated with water. You should know that methanogenesis is ubiquitous in the soil subsurface because of limestone deposits, which will release CO2, the terminal electron acceptor. You should know that toxic metals, when they're found in the environment, can never completely be degraded, but they are instead immobilized in the soil, so they won't affect water for drinking. To know the different types of immune overreactions: type 1, which is allergy and apoptopy; type 2, which involves IgG and IgM, like poor blood transfusions; type 3, which are deposits of immune molecules and proteins in certain areas of the body; and type 4, which is a T cell reaction, which is delayed hypersensitivity. You should know that the antibodies involved in infection in the bloodstream and the lymph system are IgG and IgM. Antibodies that are stimulated during allergic reactions are IgE, and that antibodies that coat the mucous membranes are secretory IgA. Should know that type 2 sensitivities are mediated by IgM and IgG. Should know that a type 4 reaction would include something like a tuberculosis skin test, that it's a delayed hypersensitivity and it does involve T cells. You should know what PAMPs are. Those are pathogen-associated molecular patterns. Okay, and that's the only term that we use for PAMPs. Okay, so next we're going to talk about the amnestic response. Remember, you have an initial response to an infectious agent, and then you have a secondary response that involves memory B cells and T cells. And the circulating antibodies primarily involved in the amnestic response are IgG. Circulating antibodies in the initial response are IgM. Okay, you should also know the different receptors, that on MHC class 1 you have CD8, and on MHC class 2 you have CD4. Okay, should also know the types of cells in the immune system. Okay, you have memory cells, memory B cells, memory T cells, regulatory B and T cells, plasma cells, which produce antibodies, helper B cells, and then helper T cells. Okay, then you have cells that are phagocytic, that are include macrophages, dendritic cells, and also neutrophils. You should also know that MHC class 1 is a protein that identifies self on all nucleated cells. Okay, you should know the difference between pyrogenic bacteria and pyogenic bacteria. Pyrogenic bacteria cause fever, and pyogenic bacteria cause pus formation. Should know the different complement pathways, classical, alternative, and lectin. And that the lectin pathway is stimulated by mannan found on the cell walls of fungus. Classical pathway is stimulated by Fc on IgG and IgM antibodies, and that the alternative pathway is stimulated by PAMPs. You should know that impetigo is caused by, in different instances, both Staphylococcus aureus and Streptococcus pyogenes. You should know that the epidermis, the outer layer of skin, is impregnated with a very hydrophobic protein called keratin. Should know that smallpox is caused by variola virus, chickenpox is caused by varicella virus, measles is caused by morbillivirus, and rubella is caused by rubevirus. Should know that the catarrhal stage of whooping cough is when you just have kind of a fever, you feel like you have a cold, and the proximal stage of whooping cough is when you have uncontrollable coughing. Should know the difference between antigenic drift and antigenic shift. Antigenic drift is just when a virus mutates slowly and it drifts, but it still stays with the same host. But antigenic shift is when you have large shifts of the nucleic acids, and it helps zoonotic diseases to transmit from things like birds to pigs to humans. You should know what a primary deficiency is and a secondary immunodeficiency is. A primary immunodeficiency would include SCID, severe combined immunodeficiency. Secondary immunodeficiency is acquired later, so that would be like HIV. Should know that the normal respiratory tract flora includes Streptococcus pyogenes, Staphylococcus aureus, Streptococcus pneumoniae, and Neisseria meningitidis. Should know that the majority of sore throats are caused by Streptococcus pyogenes. Should know how Streptococcus works in terms of degrading fibrin clots. It activates human plasmin by streptokinase. Should know what pathogen causes keratitis. And that pathogen is herpes simplex virus, and it travels through the ophthalmic nerve, and then it goes into the eye and causes keratitis. Should know that the organism that transmits river fever from the bloodstream to the eye is a helminth, actually a worm, but the infective agent is a bacterium. You should be able to look at and calculate therapeutic index for antibiotics and do comparisons of therapeutic indices for antibiotics, and always know that the antibiotic that has the highest therapeutic index is more appropriate. You want that therapeutic index to be as high as possible. You should know that the key feature of microbiology is that you're looking at organisms too small to be seen with the unaided eye. And yes, there is a doubling time calculation, so make sure that you go through the doubling time calculation given different information, given the starting number of bacteria, given the doubling time, and the total time, and the number of generations. Should know what a Kirby-Bauer test is, and what the zone of inhibition is compared to the zone of resistance and susceptibility. And that's it. That's it. That covers all the questions. Literally, what I've done is I've gone through the final exam and I've gone through all the questions. Of course, Moodle randomizes these, so you're not learning these in any specific order. And pay attention to the randomization. I hope everything works for you and that you have great success and that you're able to study hard for the final.